The idea that gut bacteria can influence the brain is no longer speculative. It is rapidly becoming one of the most transformative insights in neuroscience and medicine. But what was once a vague notion of gut feelings is now being traced with neuroanatomical precision. Scientists are mapping a definable circuit, a path by which microbial signals, born in the depths of the colon, travel through neural highways to reach the emotional and cognitive centers of the brain. This journey, from colon to cortex, is reshaping our understanding of behavior, decision-making, and mental health by showing that the brain does not operate in isolation, but as part of a deeply interconnected biotic system. At the starting point of this circuit are the trillions of microbes that inhabit the colon. These organisms metabolize dietary compounds into a rich array of chemical messengers: short-chain fatty acids, tryptophan metabolites, secondary bile acids, peptidoglycans, and microbial proteins like flagellin. Many of these molecules act on specialized gut epithelial cells, particularly enteroendocrine cells and neuropods, which function as sensors and transducers. Neuropods are remarkable in that they form synapse-like connections with afferent fibers of the vagus nerve, the body's primary conduit for gut-to-brain communication. The vagus nerve is not simply a passive wire. It is a selective, bidirectional filter. Neuropods release fast-acting neurotransmitters such as glutamate, ADP, or serotonin in response to microbial cues. This allows them to initiate electrical signals that ascend rapidly toward the brainstem, specifically to the nucleus tractus solitarius, NTS, and the area postrema, two structures that serve as integration hubs for visceral sensory input. From there, the signal radiates into deeper brain territories, not just regulating hunger and autonomic responses, but influencing emotion, motivation, and cognition. Neuroanatomical tracing studies, especially in rodents using viral labeling techniques, have mapped the flow of gut-derived vagal inputs to higher brain areas such as the amygdala, hypothalamus, insula, and prefrontal cortex. These regions are critical for fear processing, emotional memory, reward valuation, and behavioral flexibility. When microbial signals are altered by antibiotics, infection, or dysbiosis, activity in these regions shifts. For example, reduced microbial diversity is associated with amygdala hyperactivation, mirroring patterns seen in anxiety and stress disorders. Meanwhile, SCFAs like butyrate have been shown to dampen microglial inflammation in the prefrontal cortex, improving executive function and emotional regulation. Perhaps most striking is the evidence that microbial signals can modulate dopaminergic pathways involved in reward-based learning. Certain gut bacteria enhance dopamine turnover in the nucleus accumbens, the brain's reward hub, potentially influencing whether we crave high-fat foods, engage in addictive behaviors, or derive pleasure from social interaction. This offers a neurochemical explanation for why disruptions in the microbiome are frequently accompanied by mood changes, impulsive eating, and reduced motivation, a phenomenon sometimes referred to as gut-brain disconnection syndrome. These circuits are not fixed, they are plastic. In germ-free mice, the absence of gut microbes leads to underdevelopment of vagal afferents and aberrant activity in the prefrontal cortex. But when these animals are colonized with healthy microbes early in life, many of these changes can be reversed. This suggests a developmentally sensitive period during which microbial signals help sculpt the brain's emotional and cognitive networks. It also implies that therapeutic restoration of microbial signaling through probiotics, dietary changes, or vagal stimulation could help retrain dysfunctional circuits in adulthood. The implications are vast. Psychiatric disorders like depression, anxiety, anorexia, and even autism spectrum conditions are now being re-examined through the lens of gut-brain circuitry. What if some of these disorders stem not solely from chemical imbalances in the brain, but from miscommunication in the gut's neural dialogue with the cortex? What if inflammation, malnutrition, or microbiome imbalance in the colon sends distorted signals that the brain misinterprets as threat, deprivation, or emotional dysregulation? In practical terms, mapping this colon-to-cortex circuitry provides a new framework for personalized medicine. Rather than treating the brain in isolation, future interventions may target the gut-brain axis at multiple points: microbial composition, neural connectivity, synaptic plasticity, and even real-time electrical modulation of vagal tone. This integrative approach could lead to next-generation psychobiotics, therapies that not only improve mood, but also recalibrate the deep biological pathways of decision-making, craving, and self-control. Ultimately, the gut is not merely a digestive organ. It is an interface where the microbial world shapes human consciousness. And the brain is not just a processor of sight and sound, it is a neural echo chamber of the gut's microbial whispers. As we continue to trace these signals from colon to cortex, we are uncovering a second nervous system, one embedded in our bowels but fully capable of steering our thoughts, feelings, and actions. This is not science fiction. It is the emerging neuroscience of the 21st century.